Now we talk about DNA sequencing, and I will present first of all the most famous method presented by Frederick Sanger while he was working at MRC Center, Cambridge, UK. His method was known as the sequencing by chain termination. He published in 1977. So here is Frederick Sanger, twice Nobel Prize winner in 1958 for his solving the insulin structure, and then again for this chain termination method. That Nobel Prize was awarded in 1980, where he shared this Nobel Prize with Gilbert. And in Sanger, in University of Cambridge, they established an institute on his name. Now it is known as Sanger Institute. So here is the description of the Sanger method. We take the template which runs 3 to 5 direction. Actually, the sequence which we will get will be the complementary to this template. So we need a primer to begin with, since the polymerase cannot start synthesizing the strands itself, so it can add the nucleotides into some already existing chain. So this primer actually provides for a nucleotide where a new nucleotide can be added. We set up 4 reactions where we have in each reaction tube we have primers and we have all the nucleotides, except that for one of the nucleotides, say for example here for adenine, we add a dideoxy also. So dideoxy is actually the chain terminator. Rest of them they are the normal nucleotides. In the second tube we add the dideoxy for cytidine, and in the third we add for guanine, and in the fourth we add for thymine. So when the reactions they process in the first tube, we can have a situation where this nucleotide dideoxy gets incorporated, for instance at this 5th position. So we can have a 5 nucleotide long fragment. It can stop here, and we can have this longer fragment here. In the second tube we can have 3 fragments based upon the incorporation of this dideoxy cytidine. So wherever it's get into it, it can give us these 3 different fragments. The third tube give us 2 fragments, and the fourth one give us these 4 fragments. So after we get those fragments, we separate them on gel using this technique known as gel electrophoresis, where DNA runs from negative to the positive gradient of electricity. Here we have all 4 reactions in this cell. So we have this mixtures coming from tube A, C, G and T, and these fragments they run against this electrical gradient, and then their running speed is maintained by their sizes. So the smallest ones they are down there in the gel, and the largest are up there on this direction. So we see that there is a first fragment, and obviously we read them from, since they are synthesized from 5' to 3' direction. So this is our first nucleotide at 5' direction. Remember here in this slide we are not considering the primer nucleotides since we already know them. So here we have the first nucleotide which is in the lane where we have this reaction C. So it's a C here, then again we have a C, then third one which is right here is an A, and then we have T, and so on. In this way we can get the whole sequence which is the same as we started. So in this way we have got the sequence of this small fragment. Another method of sequencing was presented by Walter Gilbert and his student Alan Maxim while they were at Harvard University, and this they have different methods. So one of them which I am presenting today is which became famous, that is the DNA sequencing by chemical degradation. So here are their pictures. Alan Maxim, student of Walter Gilbert, and he was student of Abdul Salam, a Nobel laureate, originally belonged to our country Pakistan. In this method, Maxim Gilbert method, there is a chemical modification of those nucleotides, which is then followed by their degradation. And then what we do here is we label the DNA before we modify those bases. This helps in screening of these fragments. So 5' end of the DNA is labeled with phosphorus P32. Different modifications, they are done in a way that there are two groups. One group is purines and the other group is pyrimidines. So purines are A and G, and they are depurinated by formic acid. Same way, Gs are methylated. So in this way we can, first we can identify that there are purines, and then within the purines we can identify whether it's a G or it's an A based upon this second reaction. Same way, pyrimidines are hydrolyzed using hydrazine, and then once we have an idea that there we have those pyrimidines, so which one is there, we can set up another reaction where we have, we call it as T only reaction. So in that we add NaCl to the same hydrazine reaction, so that blocks the reaction of T with the hydrazine, so only C can react in that case. So in this way we can distinguish that which pyrimidine we are getting, whether it's C or T. Then there is one base modification per DNA molecule, and the cleavage is where the base is modified, and we get those different strands on the basis of that cleavage, the cleavage of the modified base. So for instance, we set up a reaction. This first one is the reaction for G, then we have a G and A reaction, now we have T and C, and we have C only. What are the advantage of setting only these four reactions? So if we see a band in some, say for instance our sequence was like this, it was G, A, T, C, G, G, A, C and C from 5' to 3' direction. The first phosphate is radio labeled, so that helps in its screening. Here in the gel we see two bands down there, and the band is present in both of them. So from this band we conclude that it's purine, and then within that we are clear that it's guanine. So if there is no band in the G only reaction, then for sure it's adenine. So here for instance we see there is just one band in G plus A but no band in G. So it's kind of exclusion plus double evidence kind of reactions where we can exclude one base, and then if we see that we have the band in both of them, then obviously we can take one of them. So here for example in T and C reaction we see just only one band, so it's not there in C but it's there in T plus C, so for sure it's T there. Here again we see two bands, one in T plus C and one is C, so we can say that it is for sure it's C. Same way we can do for others, and in the end we can have those the whole sequence that has been captured. So Gilbert methods involved a little bit complexity in terms of this chemical modifications as well as these degradation procedures. There are also critiques about the use of some chemicals which are carcinogenic, so it did not get more success, whereas Sanger method got more success in terms of its use and publicity. So modern day sequencing techniques are mainly using Sanger's method with some modifications. So there we have seen that there are different methods of sequencing which have been developed. We presented Sanger's and Gilbert's, and among them we see that Sanger method has got more success.